It's a real pleasure to introduce today's guest speaker, Cassandra Extavour from Harvard University, where she is a professor in two departments, actually, molecular and cell biology, and also organismic and evolutionary biology. And a few stages of Cassandra's career. So Cassandra actually started out at the University of Toronto, where she got a bachelor with honors. She then moved to Spain, to the Universidad Autónoma de Madrid, where she got her PhD in molecular biology and genetics. And her thesis actually was on germ cell selection in genetic mosaics. And we will see that germ cells have been really a theme that has followed her ever since her PhD, maybe even before her PhD. After her PhD, she went to Cambridge University, where she did work for a number of years as a postdoc, but also independent work before in 2007, she moved to Harvard, and she's gone through the ranks at Harvard on the faculty. So Cassandra's research interests. So on her website, it states evolution of development at multiple levels of biological organization and scale. And one focus is really still germ cells and the gonad. And those are really important cell types because they drive evolutionary processes. And she was, for example, instrumental in demonstrating that signaling and cell-cell competition are really important in the formation of gametes. It's not just all inheritance. So there's lots more going on. And I became aware of her work in 2019 when she published a seminal study on insect egg size and shape, and showed that they evolve with changes in environment and ecology rather than the rate of development. Cassandra is also a proponent of the use of non-model organisms, and we will see that today. And she spearheaded an initiative called EDEN, which stands for Evo-Devo Eco Network. And that was a collaborative effort supported by the National Science Foundation to bring these non-model organisms more into the limelight of research. And I have to admit, I'm really amazed and impressed by the breadth of the research that is ongoing in Cassandra's lab, the different approaches, the different scales, and the different models. And she's using this, or her team, to address really big and challenging questions in developmental biology. And I think they just take what it needs, you use what it takes to tackle big questions. And I think that's really admirable. Cassandra is also has received lots of awards for her research. In 2021, she also became a Howard Hughes investigator and was named the Timken Professor of Organismic and Evolutionary Biology, Molecular and Cellular Biology at Harvard. She's also a passionate and highly decorated teacher. In 2020, for example, she was named Harvard College Professor in recognition of her contributions to teaching and mentoring at Harvard. And then last but not least, I want to mention one thing which I find most impressive. Cassandra sings classical and Baroque music professionally and has a musical career as well. Apart from her career in the sciences, and in Boston, for example, she regularly sings and performs with the Handel and Haydn Society chorus. So Cassandra, I'm really inspired, and I really look forward to your talk now. And I hand over to you, and the title of Cassandra's talk is here already on the screen: Systems Approach to Eco-Evo-Devo Problems, Hawaiian Drosophila as a Model Clade. Thanks so much for being here, Cassandra.
Thank you. Thank you, Barbara, for the very generous introduction. Thank you, Michael, for the invitation and for everyone's patience in dealing with yet another virtual seminar. The seminar was scheduled quite a while ago, but of course, because of the pandemic, we've had to be flexible. So I really appreciate everyone making the time to be here today. So what I'm going to tell you about, I decided today to talk about a number of different projects that have been ongoing in the lab and are still running, that use many different approaches, different technical approaches, in some cases different organisms, to ask a large sort of integrated question, which is how we can understand how genes in one organism impact the cellular decisions of that organism, and how the actions of those genes can also be impacted by the ecology and the environment of that organism, and how that collective cohort of genes and environment working together can actually evolve over time and operate differently in different evolutionary lineages. And you'll see as I come towards the end of the talk that I'm going to talk about some insights that we have gained and hope to further gain by using Hawaiian Drosophila fruit flies as a model clade. And what you're seeing on the screen is, it's not one of our field sites, it's a view that we get on the way to one of our field sites in Hawaii to collect these flies. Now, I know I don't have to convince this audience that the expression and function of genes is critical for regulating homeostatic function of all cellular life, and also is what we look to for some clues as to what's going wrong in those processes when we have disease or pathogenic conditions. Now, my perspective on the importance of gene expression and function is that it's particularly important to recognize that even within one species, different individuals within a species, and then certainly even individuals between different populations or different species, if you want to call them that, they exhibit variation in this gene expression and in this gene function. Even genes that originally come from the same gene family, that are homologous, the exact same allele, may not behave exactly the same in different genetic backgrounds or in different environments. And so we want to understand how genes work and how they control cell behaviors using a comparative method where we compare how these things work between individuals and between lineages. And so that's sort of the premise of the comparative approach that we like to take, and it's why we, rather than using a single model organism, we use multiple model organisms, and we also try very hard to consider insights gained by other colleagues who work on yet additional models and put all of this understanding into an evolutionary framework. Now, when we're considering evolutionary change in any trait, including gene expression and function, we want to think about what is essential for evolutionary change, which of course is just defined by changes in allele frequency distributions over time. And so we know that we need to have variation for evolution to happen. If everything is absolutely identical, then there's nothing to change to. So variation is an essential part of the evolutionary process. We need to have heredity. We're concerned in evolution with heritable changes, so that the allele frequencies that are inherited are the ones that are changing across time. So we need variation. We need it to be heritable. And if we have a third element in the equation on the right, if we have differences in traits, so the variation correlated with differences in fitness, then we have the opportunity for one particular mode of evolutionary change, which is natural selection. So natural selection can operate if the variations that we see in different phenotypes also correlate with different fitnesses. But of course, as we know, natural selection is not the only mode of change. But I just mention it here because it turns out that we are quite interested in links to fitness. And so we try to choose phenotypes that we think are likely, or we can demonstrate do have impacts on fitness. And therefore, we're interested in some traits that may be subject to evolutionary change. So it's for that reason that we choose to focus a lot on the reproductive system, because it is the cells of the reproductive system that are going to pass on all of those heritable changes in their different allelic combinations. And so we consider cells of the reproductive system, both the gametes, the germ cells that are going to make, let's say, eggs and sperm in sexually reproducing systems. We're considering the somatic cells of the gonad that house those germ cells and make sure that gametes are developed properly and get out into the environment. And we like to consider also some measures of reproductive output, so some measurements of how many offspring can be generated. And you'll see us sort of quantify all of these different traits that are relevant to reproduction as I go throughout the talk. So from a developmental point of view, I see the evolutionary cycle as one whereby you segregate your reproductive propagules. We call them germ cells in animals often. So we segregate the germ cells from the soma. We differentiate different types of somatic cells so that the body can have all the different types of cells it needs, including the gonad. Through a process, which I'm just going to black box and call development, all of these different cell types get organized physically and shape-wise into their respective places and tissue types. And all of this culminates from an evolutionary point of view in reproductive output. Offspring in a new generation that will carry one set of alleles or another set of alleles. And so we're interested in traits that can impact quite directly reproductive output, because these traits will have quite a tight link to fitness and therefore potentially be of interest when it comes to natural selection. Now, because the traits that we're looking at can range from genes to cells to organisms, and recently, and we hope in the future, to the environment that these organisms find themselves in, we find ourselves doing studies that examine these many different levels of biological organization. So you'll see some examples where we've just considered the product of the gene, or we've just considered the behavior of a cell, and we try to take whatever approach we find most appropriate for whatever skill we're dealing with. We also, again, because we're looking for evolutionary principles that can help give us explanatory power across broad ranges of the tree of life if possible, rather than working only with one model organism, we often work with multiple when we also take advantage of the thousands of researchers who have come before us and look at the data that they've generated and add them to our data sets. And I'm not going to talk about this today, but recently we've become interested in how mathematical simulations of organisms, cellular behavior, tissue level behavior can also be informative in trying to uncover general principles that might inform what cells do and how those actions evolve over time. In today's talk, I'm going to be talking only about, only showing you data from Drosophila melanogaster or other species of Drosophila, so within that genus. And then, depending on the organism, the scale of biological organization that we're looking at, we will employ, and you'll see this throughout the talk, a number of different tools as appropriate. So we might be doing genetics, we might be doing microscopy or experimental embryology, we might be doing bioinformatics or other computational methods, phylogenetic comparative methods, we might be doing field work. And so we try again to employ whatever approach is, we hope, going to be most useful and most insightful for the level that we're looking at. Okay, so the trait that we're going to focus on for the next half hour or so is a trait that has to do with reproductive output in insects, which is determined in female insects in very large part by the number of a particular substructure within the ovary called an ovariole. So on the left, you're seeing a schematic of a fly, so that you can see that in the abdomen, which is down at the posterior, we have this bilaterally symmetrical gonad, and there are two sort of large lobes, which are the ovaries themselves, connected by little tubes, the oviduct, to a common oviduct and uterus. So the egg will be matured in an ovary, go down the oviduct, and exit the animal, and pick up a sperm either on the way out or once it gets outside, depending on the insect. So that's not unlike the female reproductive system in mammals. There's a bilaterally symmetrical system, and each ovary has eggs maturing inside it. But unlike the mammalian gonad, which is basically a disorganized bag of developing follicles at different stages of development with no clear spatial organization, in female insects, the ovary is highly spatially organized into distinct tubes called ovarioles, and you can see a single tube on the right-hand side. And within these tubes, eggs are maturing in a very orderly process from anterior, less mature, to posterior, that's at the bottom of the slide, more mature. And the cartoon in the middle is showing you what all the ovarioles look like, sort of lined up together inside an ovary. So what we're concerned with here is the functioning of these tubes, because they are where the eggs get made. And so eggs need to be correctly developed inside there for you to have successful reproduction. And we're also concerned with the number of these tubes, because it turns out that in many insects, including insects of the genus Drosophila, the number of ovarioles is a key predictor of reproductive output. And so that's why we're going to consider in the next half hour this particular trait, ovariole number, as a trait that I'm going to provide evidence to you that shows variability within and between populations, that is heritable, is highly heritable, and has a direct link to fitness. So the variation in ovariole number, here are some data that can back up this point. What you're seeing here is a phylogenetic relationship of 12 Drosophila species, and the total number of ovarioles per female is listed, are the numbers down on the right-hand side. These ovaries are symmetrical, and we've never detected, in the many tens of thousands of ovaries that we've counted, we've never detected any left-right asymmetry in ovariole number in this species, in this genus, I should say, in the genus Drosophila. And so if you want to know how many ovarioles a given ovary has in this species, in a given species, you can just divide the number on the right by 2. So we have the total number of ovarioles per female has quite a bit of variability. It can essentially double, a little bit more, just within this fairly closely related group of species that shared a last common ancestor maybe 50, 60 million years ago or so. So we have variability, high variability, even just within this genus. We have high heritability of this trait, and what you're seeing here are data that back up this claim. What we did was, in this small experiment, go to Hawaii, collect a number of different species, and we're showing you 5 different species here. These 5 species in the wild have extremely different environments and extremely different nutrient exposure, because they live on different types of habitats, and those habitats are listed across the top: flower, sap, etc. And what we did was we caught wild-caught females from these species. They're flying around, they've mated with whoever they did, they're however old they are, and we counted how many ovarioles those adult females had. But before we did that, because that's a destructive process, in the lab we asked them, and they agreed to lay some eggs on our food, our standard Drosophila food in the lab. And after they laid eggs, we sacrificed the wild-caught females, counted their ovarials, and those ovarial numbers of the wild-caught females are the categories called wild on the x-axis. The larvae that emerged from those eggs that were laid by these wild-caught females on our standard food now, they're not eating their sap or their fruit or whatever from out in Hawaii, they're all eating the same food as us, so the environmental contribution, the potential environmental contribution of their food to the ovarial number has been eliminated in this experiment. And all of the F1 larvae that were, all of the F1 adults that emerged from eggs laid by these wild-caught females in the lab had ovarial numbers that were significantly, that were not significantly different from the ovarial numbers of their wild-caught females. So this doesn't mean that plasticity is impossible, but it does show you that with respect to the nutrition and any other environmental factors that these flies are experiencing that are very different in the wild, the number of the ovarials that they have is very heritable and highly sort of robust to those sorts of environmental changes, at least in this experiment. So we have a trait that is variable. We have a trait that is heritable. Now I'm going to show you some information that is going to hopefully convince you that ovarial number has some relevance to fitness as well. What we see is that across different ranges of the phylogeny in insects, ovarial number can predict egg laying output. So here's an anecdotal example from our lab where we have Drosophila melanogaster females, about 40 ovarials per female, about 20 per ovary. And in our hands, one of these females will lay about 50 to 100 eggs a day. Now, Gryllus bimaculata is a cricket species that we also rear in our lab for other studies. And these females have an order of magnitude more ovarials per ovary, and they also lay an order of magnitude more eggs per day. Even within the genus Drosophila, now you might think, well, this comparison is very large because the last time these guys shared a common ancestor was a good 500 million years ago, maybe 300, but it was a long time ago. But even within the same genus, even within Drosophila, these two species, Drosophila melanogaster and Drosophila seychellia, shared a last common ancestor only around 3 million years ago. Drosophila seychellia evolved a lower ovarial number very rapidly than their last common ancestor. It only has about half the number of ovarials compared to Drosophila melanogaster, and it is laying far fewer eggs than melanogaster is. So you're seeing the number of eggs laid by a female on a given day on the y-axis, and the x-axis is showing you the first 10 days of life of females, 10 females per species in this experiment of these two species. And this is a small data set, and it's only the first 10 days of their life. But we also collected, I'm not showing you here, a larger data set where we counted the number of eggs laid per day, per female, on every day of her life until she died for 100 females for each of these two species. And Drosophila seychellia always lays fewer eggs every day. It never catches up. It doesn't live longer. So it has half the number of ovarials, and it lays half or less the number of eggs compared to melanogaster. So with this trait now that shows high variation, heritability, and fitness link, I'm going to show you data generated. 99% of the data I'm going to show you was generated by these 5 talented people, one of whom still works in my lab, and the others of whom have moved on to bigger and better things. And so I want to acknowledge the talents and hard work of these people that did the work that you're going to see in the next 20 minutes or so. So let's look at reproductive capacity from at least 3 different vantage points. We're going to look at the regulation of development. What do the cells do in order to make ovarials? And what are the important factors that control their behavior so that they can make ovarials properly and make the right number? Let's look at genetic regulation. What are the genes that are important for these cells to enact these important behaviors so that ovarial number will be established correctly in a given species? And then finally, environmental regulation. What are the elements of the natural environment in which different lineages live that may contribute to the establishment of ovarial number or the evolution of ovarial number over time? So first, let's consider developmental regulation. What you're seeing in this micrograph will make more sense to you after a couple of slides. It's a larval Drosophila ovary in the process of building ovarials. And you will learn that a number of different cell types are important for this process. Germ cells are in yellow in the middle. Intermingled cells are a type of somatic cells in green. They hang out closely with the germ cells. Blue just marks the nucleus of every cell. And you're going to learn that these cells marked in red, which are called terminal filament cells, are really important for determining not only that an ovarial gets made, but how many ovarials get made in the first place. So ovarial number is determined in the larva. During embryogenesis, a small gonad primordium is established that consists of a number of germ cells in yellow, surrounded by a single layer of somatic cells, here in white. So from that first larval instar, or L1, we molt about 24 hours later, and we get a second larval instar, or L2, in which, in this gonad, all of the cell types have proliferated. So we have more somatic cells in white and more germ cells in yellow. And they've also changed their relative spatial arrangement so that the somatic cells are almost all piled on top or anterior to the germ cells. The germ cells are occupying the posterior of the ovary. And some of the somatic cells have begun to differentiate, and that means to us they look different and they start to express genes that are different from the other cells. And we see that they are intermingling with the germ cells, and we call these the intermingled cells in green. In the next molt to the third larval instar, at the beginning of the L3 stage, or the third larval instar, we see that there's even more proliferation of all the types of cells. We see two key things that are new happening in the somatic cell population. The first is a migration event whereby you can see on the right-hand side some of the somatic cells that were in the anterior are now moving down beside the germ cells. They're on their way to the posterior region of the ovary. So there's this migration of the so-called swarm cells. I won't be referring to those too much in the rest of the talk, but I will be referring to these cells that have now begun to turn on some genes that we can notice, and they are called the terminal filament cells. So among the somatic cells, some of them now appear to take on a fate of so-called terminal filament cells. And what these cells do is they build structures that look like small stacks of coins or pancakes or whatever you like to stack, books. And these stacks are called terminal filaments. And so it's the terminal filament cells that are dedicated to making these stack structures. And these stack structures continue to be formed progressively throughout the rest of larval life, so that by the time the animal is about to pupariate, or the larval pupal transition, or the so-called LP stage, all the terminal filament cells have arranged themselves into a species-specific number of stacks. The number of stacks is going to tell you how many ovarioles the adult is going to have, because every terminal filament is the starting point for the subsequent morphogenetic processes that will divide up the rest of the cells in the ovary into the ovarioles that the adult needs to have. So that means that understanding ovariole number, which is this adult trait that impacts fitness, is in many ways a matter of understanding cell behaviors in the larval stage of this animal. Now, what Andre Green, a graduate student who was formerly in the lab, was interested in understanding is how this development process could change in different Drosophila lineages that had convergently evolved a different ovariole number relative to a last common ancestor. And by doing an ancestral state reconstruction, considering the ovariole numbers of a number of different extant species, which you can see are the numbers on the right, he inferred what the ovariole number of the last common ancestor at each of the nodes on this particular tree would have been, and those are the numbers in the yellow boxes. And from this inference, he hypothesized that the Drosophila seychellia species, with an ovariole number of only about 15, relative to a last common ancestor that it shared with melanogaster of about 30 in the bright yellow box, that Drosophila melanogaster from India, a particular population of melanogaster from India, and Drosophila seychellia, a different species that lives in the Seychelles, had independently evolved a lower ovariole number than their last common ancestor. And Drosophila melanogaster and Drosophila simulans in the same clade had independently evolved higher ovariole numbers than their last common ancestor. So this was the hypothesis that he established by doing this ancestral state reconstruction, and he wanted to know what sorts of developmental changes were happening in the ovaries of Drosophila seychellia and of the Indian strain of Drosophila melanogaster that would explain why, as adults, they had so few ovaries. He wanted to know whether or not the changes in the cell behavior program had evolved the same ways in these two different strains independently, yielding the same result of lower ovariole number, or whether there were basically different cell behaviors that could be tweaked to achieve the same final result of lower ovariole number. And I'll fast forward you to the result. He found that it was the latter. The ways that development is different in the larval ovary in the Indian strain and in the Seychellia strain compared to melanogaster from North America with the higher number, the ways that ovarian development differs are not the same between these two species, and yet they end up with a similar final result, which is lower ovariole number. So we're going to focus on these two lineages now, Drosophila seychellia and Drosophila melanogaster from India. What Andre did was to count all the cells of the larval ovary and distinguish which type was which, so he counted the germ cells, the intermingled cells, the terminal filament cells, at every stage of larval development across these three lineages. Drosophila melanogaster from North America, that we're calling OR for Oregon R, that control strain that we use, that's in the middle, that's the white bar. Drosophila melanogaster from India, which is the gray strain on the left-hand side, the gray bar. It has fewer ovarioles than normal, than Oregon R, I should say. And Drosophila seychellia is going to be represented by the black bar. and that has fewer ovarioles again than melanogaster from North America. And both India and Seychelles have fewer ovarioles than we think their last common ancestor with melanogaster did. Okay, so what he found was at the very beginning of larval development, that ovarian primordium that's established in the embryo is actually different sizes between the species. So both melanogaster species, India and Oregonor, have ovaries that are about the same size. They have about 50 somatic cells in them each. But Drosophila seychellia has only about 35 somatic cells in it. So it has a significantly smaller ovary to begin with. And remember that the terminal filament cells that are going to determine how many ovarioles you get come from the somatic cell population, not the germ cell population. So the number of somatic cells that you start with could be quite an important parameter because it might influence how many terminal filament cells you can ultimately make. Now, Andre followed these ovaries throughout their development in these three lineages. And he found that at the larval pupal stage, when they should have made all the terminal filaments they need to make, he asked what the status was of the total number of cells. Now let's focus on Seychellia first. It started with a smaller ovary, and indeed by the time it's time to pupariate, this ovary remained smaller than the melanogaster ovary. So they started with fewer cells and their proliferation rate never caught up, and so they ended up with fewer cells as well. By contrast though, we were surprised to see that the lineage from India of Drosophila melanogaster, even though it started with an ovary of the same size as its cousin from North America, by the time it was going to pupariate, it had way more cells than the North American counterpart. This was surprising to us because we know that as an adult it's going to make fewer ovarioles. So what is it doing with all of these cells, but ending up with fewer ovarioles? And it turns out that the proliferation rate difference can explain the smaller ovariole number in Seychellia, but not in India. But what explains the difference in India is the relative allocation of different cell types to the somatic cells that they do have. So the Indian lineage has far more somatic cells to work with, it turns out, by the end of larval development, far more somatic cells to work with than its cousin from North America. And yet it ends up making fewer ovarioles because it allocates a smaller proportion of those cells, of those somatic cells, to the terminal filament cell fate. So the C plot on the right-hand side is showing you the proportion of somatic cells allocated to different fates, whether posterior cells that end up posterior to the germ cells in white, apical cells, which are in between the terminal filaments in gray, or terminal filament cells, which is our population of interest, in black. And you can see that the strain from India, the leftmost bar in this C plot, has a relatively smaller proportion of all its somatic cells allocated to that terminal filament cell fate. Interestingly, Drosophila seychellia and Drosophila melanogaster from North America, they have different absolute numbers of ovarioles, but they've allocated the same proportion of the cells they did have to that terminal filament cell fate. But Seychellia ended up with fewer ovarioles because it had fewer somatic cells total to begin with. All right, so this is a sort of the classic question we're asking here is, what is the developmental basis of convergent evolution? Do we achieve the same evolutionary outcomes in the same ways or in different ways? And Andre's answer to this question was, here we have very different ways. There are so many different types of cell behaviors involved in making ovarioles and determining their number that evolution can work with any number of different sort of switches to achieve similar outcomes, whether lower or higher ovariole number. Now, what Didem Sarakaya, a contemporary student of Andre's, was interested in was whether or not these developmental processes that Andre was characterizing in lab species, could they also account for the variation in ovariole number that we see in species that are living in the wild that have much broader ranges of ovariole numbers than we see in our lab caught species? So she was the first person from our lab to go to Hawaii, establish essential collaborations with key experts in Hawaiian flora and fauna out in those islands, and to be able to collect many species of Drosophila from Hawaii, bring them back to lab, and examine their larval ovarian development and count their terminal filament cells and other cell types. And she asked whether, as in the strains that we'd examined in the lab, Drosophila seychellia and melanogaster, where we see that it's really the number of terminal filament cells that is a key predictor of ovariole number, was this the case even for naturally living species in their wild habitats that have many fewer or sometimes many more ovarioles than the lab caught strains we'd been working with? And it turns out the answer was yes. So she took a number of species that had a fairly wide range of ovarial number, and you're seeing adult ovarial number in the white bars on the bottom. And she was looking at species that had well over sort of 30 or so ovarials, or even just 1 or 2 ovarials per ovary, so a wide range of ovarial numbers. And she found an almost perfect one-to-one correspondence between the adult ovarial number and the total number of terminal filament cells found in their larvae, which are the gray bars in the histogram on the top. And so even in these naturally occurring species that have evolved a very wide range of ovarial numbers, as you'll see a little bit later on, the key parameter that is a good predictor of adult ovarial number is still this terminal filament cell population. But as you've seen in the example with the Indian strain and Seychellia, the way that you come to determine the number of cells that should be in this population can be influenced in turn by many parameters. Okay. So we've thought a little bit about some of the key cellular behaviors that are important for ovarial number establishment. Now let's think about some of the genes that might be involved in telling these cells what to do. Other people had, of course, worked on this problem, and a number of different pathways that are marked in black here had been shown to be important for signaling within and between cell populations in the ovary. The terminal filament cells in red, the germ cells in yellow, and the intermingled cells in green. And what Didem Sarakaya showed was that the hippo signaling pathway, which is a highly conserved animal signaling pathway that regulates proliferation across animals, was also an important player in regulating the numbers of these different cell types. And remember, the numbers of cell types, especially the number of terminal filament cells, is of high interest to us because it seems to be a key determinant of adult ovarial number. And Didem, through her experiments, was able to add in a few more arrows into these sorts of diagrams, the ones in blue here, showing that there are some autonomous and non-autonomous roles of hippo signaling, including Yorkie, to these different cell types to establish the correct numbers. But we knew, of course, as all of you know, that no process in development is controlled only by a single signaling pathway. And so we embarked on a number of different screens to try to find additional genes that might be operating either on their own or together with hippo signaling or some of these other signaling pathways to regulate ovarial number. And so what Tarun Kumar did, a very talented fly geneticist, was to do a sort of a classic modifier screen where he took flies where we'd induced a loss of function condition of hippo signaling just in the somatic cells of the ovary using the Gal4 system. So he'd shut down hippo signaling levels in the somatic ovary during development. Hippo signaling normally is repressing or controlling cell mitosis, and so by decreasing its activity levels, what you do is you generally increase mitotic levels. So by making flies where in the somatic gonad they had less hippo signaling than normal, they made more somatic cells than normal, they made more terminal filament cells, they had more ovarials, and they laid more eggs. These flies are happy to live just like that. They're very fit. So he took these flies, and using the US Gal4 system, systematically one by one in the somatic cells of the ovary during development, he knocked down every conserved member of every conserved animal signaling pathway, so about 800 genes, one at a time. And he looked for genes that, when knocked down, either in the context of a hippo loss of function or just in a wild-type context, changed the number of eggs laid by these flies, so he did an egg-laying count assay, and also changed the ovarial numbers of these flies. And in doing so, he found maybe about 200 or so genes that hadn't been previously characterized as having a role in ovarial number development that we think do impact either ovarial number development or egg laying or both of these traits at once. And the only thing that I want to highlight in this short time that we have to talk about this, besides the fact that, okay, now there are lots of genes that we know are involved, which we could have predicted already, even if we didn't know their exact identity, we were interested in the potential regulatory relationships between these genes. What might those be? And so we decided to, on the group of genes that we determined were causally important for ovarial number and egg laying, we decided to look at what the network topology of the predicted protein-protein interactions between these gene products would be, based on existing databases from others on protein-protein interactions that have been done genome-wide. And basically we found two important things out of this network topology analysis. One was that there are a core group of genes that were within the hits that we got from the screen that regulate both egg laying and ovarial number that are more tightly connected to each other in terms of predicted interactions than you would expect by chance from any random group of genes in the genome. And we'll call this the so-called core module of genes. And the second thing that we found is that we find other, we're calling them tentatively submodules, smaller groups of genes that again are more tightly connected to each other in terms of protein-protein interactions than you would expect by chance, and that together all share in common the fact that they regulate a particular specific phenotype. For example, they regulate ovarial number in a hippo loss of function background, or they regulate egg laying in a hippo sort of normal function background. Or so we found sort of 6 submodules which are predicted sort of small gene regulatory circuits potentially based on protein-protein interaction that can regulate individual phenotypes, as well as a so-called core module that appears to regulate all phenotypes. So when we think back to the findings from Andre's study about the convergent evolution of lower ovarial number by different developmental processes, we were intrigued to see this suggestion of submodules of little genetic networks that could separably regulate potentially individual phenotypes. All of the phenotypes are relevant to fitness, but seeing that the structure of genetic regulation can sort of turn or manipulate individual elements of this fitness equation, and each individual element, of course, may be determined by some specific anatomical structure or developmental process or metabolic process that collectively are all needed for development. Seeing this substructure of separable genetic modules gives us a sort of a genetic regulatory intuition for how it is that evolution can essentially choose from among many, many different knobs to turn when it is intending to raise or lower ovarial number in response to whatever the natural selective pressures might be there. And finally, I'll just leave you with this one specific candidate gene that we became very interested in, which is a sort of a candidate signaling pathway, which is insulin signaling. You might remember that in Drosophila seychellia, we find half the number of ovarioles compared to melanogaster. We find it starts life with a smaller gonad. It has fewer somatic cells in it, and those cells don't proliferate as quickly as in melanogaster, and you end up with a smaller larval ovary, fewer terminal filament cells, and fewer adult ovarioles emerge. What Andre wondered was whether or not, basically these differences in phenotype between the two species reminded him of differences in phenotype between wild-type melanogaster and insulin signaling loss of function mutants in melanogaster, which also have fewer ovarioles and, as Andre confirmed, fewer cells in their ovary right from the beginning and a slower proliferation rate of those ovaries. So he thought to ask whether or not there might be any heritable differences in insulin signaling levels between these two species. In other words, could evolution of insulin signaling levels between species be in part responsible for the evolution of different ovarial numbers between these two species? And he tested this hypothesis in a number of different ways, and all of the tests that he did yielded results consistent with this hypothesis. And I'm just showing you one of his results here, where he quantified the amount of phospho-AKT, which is a downstream effector of insulin signaling. He quantified the levels of phospho-AKT in the larval ovary at the time of ovarial number determination in the two species. And you can see that in the melanogaster ovary in black, there's significantly higher levels of insulin signaling operating when it's time to make that key decision of making ovarial numbers than there is in Drosophila seychellia, which is the white bar. So at the moment, we have a working hypothesis when it comes to this particular species comparison, which is that there have been heritable changes in members of the insulin signaling pathway in the lineage leading to seychellia that lead to this fly having a lower systemic level of insulin signaling, which has a number of different impacts on its phenotypes, including a lower ovarial number. This is a working hypothesis that we're still in the process of testing. All right. Now, finally, I want to tell you in the last few minutes about some of our work thinking about how we can incorporate the reality of huge amounts of environmental and habitat-specific differences in ovarial number, and how we might begin to understand how different environmental factors are impacting this trait. So the studies I've showed you so far, and most of what we've done, has been with laboratory-reared populations of Drosophila species that are from a number of different continents, that have a range of ovarial numbers between about 10 and 20, roughly, per ovary. But we know that in the Hawaiian archipelago, on these islands, a rapid adaptive island radiation of Drosophila species took place over the last 25 million years, resulting in about 1,000 different species of Drosophila, which is a lot because worldwide there are only about 4,000 species described for this genus. And among these flies that evolved in Hawaii endemically, they have a massive range of ovarial numbers, a much greater range of ovarial numbers than we see among the remaining 3,000 species that are all over the world. There are flies in Hawaii, Drosophila flies, that have as few as just 1 or 2 ovarioles per ovary, and flies that have almost 100 ovarioles per ovary, which you won't find outside of Hawaii. And so for these reasons, DDEM was very interested to examine those wild populations there. These flies are interesting in many, many different ways, not just their ovarial number range. They also have evolved repeated specializations and diversifications of their preferred habitats, and these are the substrates where you will find the flies. This is where the flies lay their egg. That means that's where the larvae hatch. That means this is what the larvae are eating. And that larval nutrition is key because it's during that larval stage that ovarial number is being determined by proliferation of terminal filament cells and so on. And so what Sam Church did, a recently graduated student, was to construct a robust phylogeny for the Hawaiian flies and to use some evolutionary modeling to infer what ancestral substrate conditions might have been based on a huge amount of literature that had previously documented what types of substrates these flies live on, and that's what the gray and black bars on the right of the tree are. They reflect the rearing records that have been recorded for these different habitats for different flies in the literature. And everywhere you see a circle on the tree is where we infer that an evolutionary shift occurred where the flies evolved a preference for a new habitat, and the different types of habitats or food substrates are the different colors that you see on the tree. So we have a very dynamic evolutionary history of shifting substrate types. Doing this work may sound, very exciting and sunny and glamorous. Actually, it's just going into the forest and looking for maggots among rotting vegetation. So it's not as glorious as it might seem, but we do find it worthwhile. Just to remind you something that I just said, Hawaiian Drosophilids have undergone a massive adaptive explosion in a very rapid period of time. And combining the phylogenetic information with the known age of these islands, which have been recently formed by volcanic eruptions and continue to form as well, we have some sense of when different species arose and which islands they first arose on. And most species are found on more than one island, but on these different islands, they still retain, in most cases, their habitat specificity. They prefer this flower or this type of leaf, etc. And so we like this study system, this so-called sort of model Clade, and our hope is that going forward, it's going to provide a beautiful comparative system where we have a robust phylogenetic framework, a massive amount of morphological and ecological diversity to speak to, and so we're going to be in the process of working with others to generate more and more genomic and functional genetic resources to be able to work with these flies. Catching them is hard work. They literally have to be caught one at a time, either with nets or with fly suckers from their different substrates and from traps that we set for them. But some of them in our hands have been, and in the hands of many others with more experience than us, are happy enough to live in the lab with slightly modified setups, and we hope that we can develop some of these into more tractable laboratory systems in the future. So before I go into the final section, I want to engage in a thought experiment with you because we're going to consider differences in gene expression between different species of Hawaiian Drosophila. And we're going to be hoping that this will lead us to changes in gene expression between these lineages that are responsible for the differences in ovarial numbers that we know exist between these lineages. The problem is, these are different lineages. They've been evolving to be different from each other for millions of years. Not that many millions, maybe a couple tens of millions, but still a long time. We don't expect their gene expression to be identical across the board. So how will we know which of the differences in gene expression that we know we're going to observe are relevant to the trait we're interested in? Okay, it's not a trivial thing to figure out. And so I hope you can appreciate that with this little thought experiment. Let's say you're interested in the expression of a particular transcription factor that you love, and you're interested in the sort of ancestral state of the expression of this transcription factor. So you assess its transcription, and it's the stage at which it is expressed in a number of different taxa that you have access to in your lab. So you pick these 3 insects, and you see what stage it's expressed at, and you see in 2 insects it's expressed at one stage, in a different insect it's expressed at a different stage. Okay. Based on sort of strict parsimony, sort of the smallest number of changes in expression timing, based on that framework, you would conclude from this taxon sampling of 3 that the gray state being expressed at one stage is the ancestral state. It's more common, it's in the lineage that branches most close to the base. You would say the ancestral state is the gray expression, and the derived state is the blue expression, or the second stage, or whatever it is. This is not an uncommon scenario to find yourself in as an Evo-Devo researcher or as somebody doing comparative work, because you're typically not assessing gene expression in a spatial way or even in a temporal way in thousands of species, right? And so often your sample size is very low when it comes to comparing taxa. Sometimes you even only have 2, okay? The problem is that if we're sort of honest with ourselves, we know that there are a lot more species out there that we haven't sampled. And so for strict parsimony to be correct, what you're really also inferring is that change in the timing of expression, that's the trait we're considering in this experiment, change in the timing of expression is super rare. In fact, it evolved only once. And I happen to get lucky enough that in my sample of 3 species, I got the only one among the over 1 million described insect species, if we're talking about insects here. However, you say, okay, well, maybe I think it's unlikely that this change evolved only once and that I happen to be so lucky, because by chance, if it evolved only once, I wouldn't expect to get it if I just picked three animals at random. So maybe it did evolve more than once, and that's why I got lucky. It evolved sort of frequently enough that even just by sampling 3, I could get it. But still, I don't think it's that common. And so maybe my conclusion is still right. But if you think that it evolved often enough so that by chance, if you sampled only 3, you should still pick it up, well then, considering that we've got 1 million insects and 500 million years of animal evolution, it should actually have evolved many, 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 many more times than just once or just 3 or 10 times. So there should have been a lot of changes. Again, if by chance our sampling of only 3 is truly to be representative of what happened, then you need to invoke a lot more changes in a sort of sample size of 1 million there, so that when you pick 3, you actually just get 1. And in this case, you begin to realize that trying to apply strict parsimony is not going to help us here, because we have too many unknowns. And if, let's say, you have information on the evolutionary rates or the effective population sizes of these species, and you realize that actually evolutionary change in any of the many, many, many nucleotide sequence changes that could have occurred to give you a change in expression timing of your gene of interest is extremely likely, then essentially by sampling 3 organisms, you have no idea, you have no basis to infer what an ancestral state was. So this is a serious problem that we face when we are doing comparative studies, when we want to not just compare things and say how they're different, but try to infer which differences are relevant to traits that we are interested in. And so Sam is thinking about how to do this, and this is an approach that he took that I'm sharing with you, and this is in review right now, and so we're very open to any feedback on how we might think about this. He went to Hawaii, he collected many species, he generated RNA-seq libraries, bulk RNA-seq libraries from a number of different tissues of interest, from individual flies, the ovary, the head, because the head is controlling behavior in a lot of ways that we hope we're to be interested in later, and then he used the carcass for sort of a whole body gene expression library. So he's got 12 different species and 3 individuals per species, all females. And we also have males, but I'm just talking about the female data here. And so he can assess the differences in gene expression between these different libraries, of course. And so in this one particular comparison, down the y-axis are just the different species that he sampled. And here he's comparing the differences in expression levels on a log scale, which is on the x-axis. And this plot is identifying genes that have a higher expression level in the ovary of a given species, and genes that have a higher expression level in the carcass relative to the ovary of different species. And then the colors that he's overlaid are whether or not these are genes that are highly expressed in the Drosophila melanogaster ovary, because of course the existing public databases give us all this information for melanogaster. And what he can see, what this plot tells you, is that most of the genes for most of the species that he's looked at, most of the genes that have a highly enriched expression in the ovary of these Hawaiian species are also genes that are highly expressed in the melanogaster ovary. Okay, so Drosophila ovaries have high expression of a lot of genes, and many of them are the same across species. That's what we're seeing. Okay, great. But which of these genes are the most interesting to us? Which of these genes do we think might have something to do with the differences in ovarial number across these species? We can't tell just based on the fact that they're highly expressed in the ovary. Well, maybe we can ask what differences in ovary bias gene expression we find between lineages, okay? And so what he did here in this plot, we're looking at a different number of different branches in the phylogeny. So the phylogeny is the tree on the left, and each different branch is shown with a number, and those numbers are reflected in the x-axis of the plot. And so each x-axis is basically a different sort of species and node. And he's marking in red genes that in that lineage have evolved a higher expression in the ovary than their ancestor had. And the blue dots are showing you genes that in that lineage evolved a lower expression level in the ovary, or a bias towards the rest of the body compared to their last common ancestor. Okay, so this shows us not just what genes are highly expressed in the ovary, but what genes evolved a high level of expression in that ovary, in that lineage. Okay, But still, how do I know which ones of these might be relevant to any differences in ovarial number between the species? There's no way to know for sure, of course, without functional testing, but we want to narrow it down because this is too many genes for us to test one at a time. So we're looking for example, are there groups of genes that evolved sort of unusually high changes in ovarian expression? That's one way to look at it. Are there groups of genes that together evolved a higher expression level in an ovary? They went along together at a greater rate than you would expect by chance. And most importantly, are there genes that have done this more than the other genes in the system? All the genes have evolved changes in expression, but are there groups of genes in the ovary that all together show a higher correlated rate of increased expression in the ovary? Perhaps these might be genes that are evolving in a different way compared to the background level of expression level change, which we know is going to happen because they're diverging lineages. Maybe these might be candidates for further study. And so one way to try to represent this is to ask how much of the difference in expression between two data sets can be attributed to the fact that these are very distantly related species. So variation across species is the blue plot here. Or how much of the difference in expression between samples can be attributed to differences not between the species but between the ovaries. So in other words, these species might be really different, but their ovary expression look really similar. Or these species are very far diverged from each other in evolutionary space, but their ovarian profiles are very close to each other. And so if we compare sort of how much of the variation in gene expression, that's the y-axis, can be attributed to evolutionary distance, that's on the x-axis, what we're looking for are genes in the top left. Genes with a big difference in expression levels, but pretty close together evolutionarily. And so that big difference in expression levels is more due to a change in the tissue level, a change in the ovary specifically, not just because these species have been separated by a long evolutionary distance. And so looking at genes, for example, when we compare genes from flies and clades A and B, might be a good place for us to start doing our comparisons, rather than, for example, comparing one of the species right at the bottom of the tree with one of the species right at the top of the tree, because this analysis is suggesting to us that those species have diverged so far that you won't be able to distinguish what changes in ovarian expression levels are really important to the ovary and which ones are just different because these species are now extremely different in all ways. I hope that makes sense, but this is one of the ways that we're considering the data that we're getting from these Hawaiian Drosophila so that we can further narrow down our functional studies in the future to genes whose changes in expression levels or other expression features we think might be actually relevant to our trait and not just the result of diversification of the lineage in general. So I'm going to, this slide, I'm going to skip it in the interest of time, but I'll briefly tell you that what we've done is taken these groups of genes that have evolved great differences in tissue level, they've evolved great differences in tissue level expression, even though the species haven't diverged very much. And we've asked whether among those genes, their Drosophila counterparts are more likely to interact physically or genetically than we would expect by chance, and it turns out that the answer is yes. So this is further support for our hunch that looking at these genes that have evolved more differences between tissues than they have between species, and that their counterparts in melanogaster show unusually tight linkage in terms of physical or genetic regulation, these might be the candidates that we want to look for that might have evolved differences in their expression or function, causing changes in ovarial number in these different Hawaiian species. So to wrap up, from these different approaches, we think that we can conclude that adult reproductive capacity in this particular system relies a lot on some specific cell behavior choices that are made much earlier in life during the larval stage. There's no single smoking gun signaling pathway that controls these important cell behaviors. All animal signaling pathways are involved in this. And so all of these pathways and their target genes could be targets of evolutionary change for this trait. Nutrient sensing molecular mechanisms like the insulin signaling pathway may be links to the heritability of ovarial number and the plasticity of ovarial number. Doing thought experiments and some simulations that help us establish null expectations for how much change do we expect to see when we do comparative studies can provide us with a useful framework to move forward when we look for the genetic basis of evolutionary change in natural populations. And finally, we know that broad taxon sampling is still important. It wasn't enough just to compare a few species in the lab that had diverged fairly recently. Going out to wild populations that occupied very different habitats gives us more information about the evolutionary process. So I want to thank you all for your attention. Thank, of course, my very interdisciplinary lab that makes all this work possible. And I'd be happy to take any questions that you might have.
Thank you so much, Cassandra. That was really very interesting. And I open up the floor for questions. Are there questions? Mark has already one. Mark, go ahead.
Hi, yeah, sorry, I forgot to unmute myself. I really enjoyed that talk. I was just wondering, because you find both insulin and hippo signaling as regulators of the variability in a variable number.
Yeah.
Do they change? So have you looked across evolution for those, and do they change in a manner that's related or different to body size overall? Because both of them should control body size and cell number.
That's an interesting question. So I can tell you that I don't think we've done exactly what you're asking, but we've done things that are related. So one thing that we did was look at the relationship between ovarial number and body size in the Hawaiian Drosophila. And many others have done this with sort of non-Hawaiian Drosophila from the other parts of the world. And under optimal nutritional conditions, most studied lineages, so it's mainly Seychellia, Melanogaster, Simulans, Yakuba, the closely related guys. Under optimal nutrition conditions, there can be a relationship between body size and ovarial number, but when they're starved or under different types of nutritional conditions, sometimes that relationship breaks down. And what we saw in the Hawaiian flies is that ovarial number and body size go together in certain groups, in certain clades, but not in others. And the correlation was not the phylogenetic relationship, but it was the substrate type. So the species that had evolved specializations on leaves and fruit and bark had evolved smaller ovarial numbers convergently when evolving specializations on these different habitats, but their bodies stayed large. But then the flies that have evolved their specializations on the very abundant, more general substrates like bark, sap, they have a body size ovarial number relationship that scales quite well. That's pretty linear. So at least in those Hawaiian flies, evolving a specialization on a less abundant food type correlated with a drop in ovarial number, but no change in body size. Yeah.
Maybe I can ask a question along the lines with the insulin signaling. Do you know where this increase actually happens, in which type of cells?
Yeah, we don't know. We don't know. We know that the sort of 2 and 6 ILP producing neurons in the brain are important for ovarial number determination. So if we kill those neurons, the ovarial number drops. So ovarials respond at least to ILPs 2 and 6, but I don't know about the other. They might respond to the others as well. We found a bizarre result that we did not follow up, and I don't know how to interpret it, which is that, we'd started off with the simplest idea, which is that some ILP produced in the brain makes its way in the hemolymph to the ovary, okay? And that's part of the situation. But I don't know why he decided to do this, but Andre tried to shut down, he used RNAi against the ILPs in the ovary. We don't have any evidence that they are producing ILPs. We haven't looked directly. It would be great if somebody would look. But when he used RNAi against some of the ILPs in the ovary, he also changed ovarial number. And it could be that, like other tissues, they are not only receiving some systemic ILPs, but perhaps also producing some of their own ILPs for use in who knows what sorts of situations. And that's a process that we perturbed with that. I mean, I would love for somebody to work on that. But we don't know. So, but I suspect that both systemic and sort of within organ sources are probably used by the ovary, as other organs do.
Yeah. Great, thanks. Jonas.
Yeah, hi, thanks for the talk. I don't really know much about evolution, but I have an evolutionary question, so it might be a little bit naive, I don't know. But so you kind of pointed out, I don't even know if I can phrase the question, but you kind of, so the nice thing about the ovaries is the link, the sort of direct link in a way that you get between the fitness aspect and sort of the variation and the organ itself. So I'm wondering, do you expect that evolutionary pressures themselves are in some way systematically different for organs that are more sort of gonad related versus somatic organs, if you will. Like you might imagine that it's like what you want is just like high robustness and high efficiency of production, whereas in other things you would favor diversity, adaptivity, that kind of stuff.
Yeah, that's really interesting. That's really interesting. I think, you know, if we didn't know about development, then I might say yes, right? Like if we thought that cells and organs were just like Lego blocks and we could just put them in and one block doesn't impact the other block, except we know now that that it basically everything is connected to everything, and any signaling pathway that you alter because your wing is bigger or your olfactory receptors or whatever is almost certainly going to also impact ovarial number because every pathway is involved there. So because of pleiotropy, it's not obvious to me at all whether ovarial number is even a direct target of selection in any system. It certainly has all the features that it could be. It's going to impact fitness, right? There's variability, but there are many other factors that could be impacting in this case, the best correlation that we see for evolutionary change of ovarial number is habitat type, right? But is their fitness increased on these habitats because their ovarial numbers change, or are they evolving the ability to deal with these habitats by altering whatever other aspect, and whatever genes are needed to change those aspects are also changing the ovarial number, right? What's interesting is that when they evolve specializations, they often drop ovarial number. They don't increase it, even though many of the specializations that are documented are where the fly evolves specialization on a particular fruit or a plant that's toxic to most other systems, but the fly evolves some new enzymatic processing to break down the toxin. And what it ends up having are fewer ovarials, not more. And you could make an argument about how making eggs costs a lot of money, and so it's costly, and so then they don't, I don't know what currency these fitnesses are traded in is a problem, right? But I think that my intuition would be that because of the pleiotropy of development, we wouldn't necessarily expect reproductive systems to sort of evolve in a particularly targeted way, because they're going to be dragged and can drag as well other organ systems with them. And it's not obvious to me that the fitness output is going to be the first determinant of evolving a specialization or occupying a new niche. It's going to be, can you physically get there? Can you eat the food that's there? And however the reproductive system adapts to it, once you're actually alive there, then you produce what you can produce. So
maybe as a quick follow-up to that on the pleiotropy question, can you tell whether like how much or which fractions of the genome effectively are specific, right, to ovarian development? Let's say like they are these parts are only active or are only related to maybe there's some specific transcription factor in terms of fate specification or some epigenetic modification that makes them unique to ovaries, and therefore you can independently evolve from other things, or is like everything coupled?
The only things that I know of, without thinking about it too deeply, the only things that I know of that are definitely just specific, are programs that are involved in gametogenesis, right? And even among those, like the genes that specify germ cells, most of them also have some other job in some other cell type sooner or later. And so the genes involved in meiosis, there's a handful of those that are really specific to meiosis, and to my knowledge don't have other jobs elsewhere. But so yeah, actually making an egg or a sperm, there are a handful of genes in those systems that you're only going to find in those systems, but pretty much everything else sooner or later has some role elsewhere. So it would be hard to have zero impact elsewhere.
Interesting. Thank you.
Yeah.
I think in the interest of time, I know Cassandra will be meeting with Mark right away. Yama, can you ask your question later? I think you're meeting with Cassandra as well.
Yeah, sure.
Yeah, good. I don't want to cut Mark's time too short, but that was really fantastic. Thank you so much. And thanks everybody for coming. And I hope you will enjoy talking to some of us now.
Great.
Thank you, Cassandra. Bye.